let's make some gene lists. Here's what I want you to do is I want you to go to Edit, go to Clone Spreadsheet. And you can name it whatever you want. Let's make 3 of those. So I'm going to go OK. Select your original one again, or you can select that one, it doesn't matter. And go Edit, Clone Spreadsheet. Okay. Yep, call it 1. And then we'll do it one more time. It's not making me make a clone of a clone. There we go. Okay, does everybody have 3 spares? These are going to be our gene lists. Let's go to the first one. I am interested in genes that change at both, that are different between my 2 different cell types, pluripotent versus embryonic, at both time 0 and day 5. So again, we did those contrasts. So what I want you to do is go to that contrast that gave the EPS cells at day 0. Oh, and you can see here, here's our mean at gene expression. Here's the p-value, and then it'll give you fold change. So this is whatever value is on the top. So this is all expressed at versus IPS values. So for this top gene, it was about 6 times lower in the IPS versus the ES. Okay, here's what I want to do is I'm just going to take, since everything's so significant, we're getting a lot of different genes. What I'm going to do is I'm going to take the top 1,000 of this. So I'm on column 13, right? Sort by p-value. Sort ascending. Again, these are all the genes that are different at time 0 between our pluripotent and ES. Go down to, so on this bar here, go down to row 1,000. And you might have to expand this column so you can see it. Put your cursor on row 1,000, hit shift and select it. Now right click and say filter include. Everybody do that? Okay. Now let's save this real quick so we know what it is. So go to File, Save as a text file. Make sure that study is highlighted that we just did, the one with 1,000. Go save as text file. And you can save this. It'll say filters are applied. We go, yes, we still want to save it. That's why we made an extra copy. So wherever you're putting this, go say, this is day 0. And then I go IPS versus ES. And I usually go underscore and then top 1,000. And then I go sig maybe. Hit save. Everybody with me? Anybody having problems? No? Again, this is why I do YouTube. Okay, let's do the second one. Okay, so we did day 0. Let's go day 5. Let's find out all those genes that are different at day 5. Where did I put it? P-value 5 iPS cells versus EPS cells. P-value. So it would be column 17. Right-click, sort ascending. And again, do the same thing we did for that other file. Let's take the top 1,000. Yeah, you'll notice as the course continues, I don't have a very high opinion of p-values. I'm working on developing another way to look at data. I hate people that just, hug p-values. But where's your p-value? Yeah, don't worry about it. All right. Again, select, right-click, filter include. So you should see rows 1,000. Again, let's save this file, save as, text file. Yes, I know I have filters. That's the point. Okay, so here, I just selected my other, basically what I called the other thing, except instead of day 5, or day 0, I'm going to put a 5 there. Save. Trust me, there's a method to my madness. We're actually going to do a Venn diagram, which is awesome, which is one of the things Partech does really, really well. Okay, let's go to our third one. What should we compare to these two things? What do you think? I'll give you a hint. It's going to be a top 1,000 significant things, but what test do you think? We have all the genes. We basically found the top 1,000 genes that change at the different cell type at day 0, the top 1,000 genes that change at day 5. Now what do you think we should look at? Any ideas? I don't hate him that much, but actually what I was thinking about is, so we could do day 5, 0, and 5, but there's just so much difference there. Here's what I want, is I want to find those. So we found things that are different at both states, but we really haven't found a difference between day 0 and day 5, right? Do the combination. So under p-value and cell type, so that'd be column 8, sort that ascending. Okay, same thing. Go down to row 1,000. I can find it. There it is. Okay. Shift, filter include. Okay, now save this as file, save as text file. Yes, I know it's filtered. Okay, so in this case, this is going to be, so this is an IPS versus ES, so I would say this is going to be day, I don't know, I'll go and cell type. You can call it whatever you want. Again, you'll notice, though, I tend to try to put as much information in the title as possible. Especially me. I take the worst lab notes known to man. In fact, I don't. So, actually, I do. I put my notes in the titles. So hit save. All right. So now we have 3 different lists. If you look at, here's the deal. So I took the top 1,000 because I kind of want an equal comparison. And I'm not too concerned because if you look at probably our least significant effect is the day and cell type together. But if you look at our last gene, what's the p-value there? 0.007. Okay? If I was looking at a PC, a PCR or some Western, they would be ecstatic for this p-value. So pretty much these genes are different. It's just that now we have equal numbers of difference. Now we can kind of compare to see what's going on. What do you expect, right? So again, we're taking genes that are different at day 0 between our 2 cell types and different at day 5. Do you expect a lot of similarity between those lists? No? I don't know. Let's find out.